Hi, yes, I'm Dr. Philip Pearl, the Director of Epilepsy and Clinical Neurophysiology and William G. Lennox Chair in the Department of Neurology at Boston Children's Hospital and Professor of Neurology here at Harvard Medical School in Boston. I also have an appointment at the Berklee College of Music and the Music and Health Institute. So I have a fairly diversified career, and my principal research area for a long time has been in the inherited metabolic epilepsies, with a particular emphasis on the pediatric neurotransmitter disorders. A large group of those disorders that I've been studying for decades has to do with GABA metabolism, gamma aminobutyric acid, which is the major inhibitory neurotransmitter of the brain. It turns out today we're talking about a different neurotransmitter disorder, but also within the large category of pediatric neurotransmitter disorders that I've been studying for years. At this time, we're talking about the disorders involving dopamine and serotonin, often called the monoamines. These are very important neurotransmitters. As you know, folks recognize dopamine as an integral neurotransmitter. There are multiple pathways in the brain. There are actually 5 different pathways that are dopaminergic. And if there's one that's best known, it would be in the motor system, especially the pathway that goes from the midbrain or the substantia nigra, where dopamine is produced, into the basal ganglia, particularly the putamen. And folks are used to hearing about Parkinson's or Parkinson disease. It was really described by James Parkinson. He didn't have it. He was a neurologist who described it, so it's called Parkinson disease. And this is a disease generally associated with older age, where there's loss of dopaminergic neurons in the basal ganglia, in the putamen, and a person develops so-called Parkinsonism, the stooped over posture, the lack of facial expressivity or so-called hypomimic facies, the resting tremor that's well known, even a postural instability where there is a predisposition to falls. So that's Parkinson's disease in the adult. It turns out that the synthesis of dopamine is an important biochemical pathway that can be abnormal as a congenital inherited metabolic disorder, which is right up my area of the inherited metabolic epilepsies that cross over to the inherited metabolic movement disorders. In other words, the pediatric neurotransmitter disorders. And the article that just recently published from an international consortium in which I am one of the US participants has to do with a disorder that you might consider as congenital Parkinson's disease. It's literally babies and infants who are born with an inability to synthesize dopamine. So it's almost like presenting with Parkinson's disease or Parkinsonism as a neonate, or more typically as a young infant. And this disorder is related to the deficiency of an enzyme known as aromatic amino acid decarboxylase. So we're going to just call it AADC deficiency for short, but that stands for aromatic amino acid decarboxylase. Now, years ago when I was in medical school, we called it dopa decarboxylase because it was pivotal and is pivotal in the transformation of L-dopa to dopamine. Also, the same enzyme that we used to call dopa decarboxylase converts 5-hydroxytryptophan to 5-hydroxytryptamine, which is serotonin. So now the name has been changed to aromatic amino acid decarboxylase because it synthesizes both crucial reactions, and especially the synthesis of both dopamine and serotonin. Obviously, another very important neurotransmitter, well described as important in mood disorders, right? We talk about SSRIs, selective serotonin reuptake inhibitors used for depression. So here we have a biochemical disorder that affects the synthesis of 2 of the most important neurotransmitters, dopamine, that's important with movement, as well as mood and learning. And of course, dopamine is synthesized into the catecholamines, epinephrine and norepinephrine, so important centrally and peripherally. For example, with the autonomic nervous system, the adrenergic receptors, the fight or flight response. So we're talking about the synthesis of dopamine, epinephrine, norepinephrine, and serotonin. All these are abnormal in this condition called AADC deficiency. Now, remember I said AADC stands for aromatic amino acid decarboxylase. As you may remember from biochemistry, all decarboxylases require a particular vitamin, Which vitamin is that? B6. Pyridoxal 5-phosphate is the biologically active form of pyridoxine. We get pyridoxine in the diet. We have some endogenous pyridoxine, but basically it all has to be converted into pyridoxal 5-phosphate, and that's vitamin B6. As you may know, there's a vitamin B6 dependent epilepsy syndrome. The B6 or pyridoxine, just like folinic acid or folic acid, is so involved in well over 150 reactions in the central nervous system. So we're talking about a disorder with protein manifestations, specifically an enzyme deficiency that also is dependent on vitamin B6. The particular paper that came up that was just published is the latest report really surveying the number of patients worldwide that have been diagnosed with AADC deficiency and the mutations or the variants in the DDC gene, the dopa decarboxylase gene. That's the gene involved. This disorder is inherited as an autosomal recessive disorder. Patients are either homozygous for pathogenic variants or they're compound heterozygotes, meaning they have different mutations on each allele from each parent, but by having both of them, it acts as an autosomal recessively inherited condition. So the parents are obligate carriers, the siblings may be carriers, but the affected children are either homozygous or compound heterozygote for pathogenic variants of this disease. And this paper clarifies and updates the number of patients that are now known to be identified in the world, which is approximately 350, and the frequency of the variants. The most frequent variant, of course, is well known to be a variant in Taiwan due to a founder effect. And the disorder is much more common in Taiwan and to a lesser extent Japan than anywhere else in the world. And that's where a lot of the initial work was done on this disorder, including the first trials of gene therapy. But part of the emphasis on assessing the prevalence of this disease and the genotypes are to understand how the genotypes affect the protein, which was the work of this study, and what's happening now in metabolic disease, and the fact that gene therapy is right here with us on the horizon, and actually recently approved by the EMA, the European Medical Agency, for this condition. Here at Boston Children's Hospital, we have already done a case of gene therapy for this disorder on an experimental basis. This study is timely because we have gene therapy at hand. And the gene therapy is actually intraparenchymal. It's not given through an IV. It's not given through intrathecal through, say, a lumbar puncture to place the substance in the subarachnoid space, as in a spinal tap, but it's given directly into the brain. There are two approaches. One is in the midbrain itself, in the substantia nigra. The other is in the putamen, one of the key nuclei of the basal ganglia. So this study establishes several key things. One, the prevalence of this disorder. There are probably many undiagnosed cases, but 350 patients have now been described. Many previously reported, and an additional set of new patients was identified in this particular study. The next thing is it's a different approach now to the gene variants. We used to say were mutations. Mutation is actually the process of the change in the gene producing the variant. So now we tend to use the terminology variant. Sometimes you'll hear that used interchangeably, the mutation of the variant. But this paper gets at a key product of this international consortium that we have in AADC deficiency to try to determine which of these variants are pathogenic, definitely pathogenic, probably pathogenic, possibly pathogenic, probably benign, possibly benign, or benign. The American Academy of Human Genetics and all the genetic associations really throughout the world are very focused on trying to define the effect of a variant. Because some variants are benign, they run in the family, other family members are normal. We call that a normal polymorphism. Some variants are unique to that patient. No one else in the family has it. The patient has a unique phenotype. That is almost always a pathogenic variant causing disease. This study uses a new approach of looking at the variant and using in silico methodology, meaning computer-based methodology in silico, we say, kind of like Silicon Valley, not really in vitro or in vivo, but in silico, where the effect of the variant of the DNA on the protein is estimated to decide where or how the variant affects the protein. This protein happens to be a homodimer. And by looking at in silico computer programs, one can look at the variant and say, oh, this changes the amino acid composition, or this does something to the protein at the binding site for pyridoxal 5-phosphate or vitamin B6. Or this variant affects the composition of the protein that changes the folding of the protein. Or this variant changes the homodimer, so it's now a heterodimer, and one aspect of the protein looks totally different than the other, and they're not going to work as well. And the whole idea, and this is now being spread to a wide range of genetic disorders, is to predict through in silico programming how the variant affects the protein product of the DNA, and therefore how it affects function. Traditionally, we would rank a mutation or a gene variant in terms of pathogenicity based on how well conserved it was. In other words, was this a highly conserved area that runs through the genome of all humans and other species, and therefore is highly conserved? It's a really important area of the protein versus is it not so highly conserved, and therefore the human being can withstand variability in it. So how highly conserved is the area? And also, it's kind of confusing because it's almost like a misnomer when we say conserved, because there's also whether it involves a highly conserved area in a different sense of the DNA and the ultimate protein, meaning can the substitution of one amino acid make a major change in the protein, or would it be a relatively minor change because this amino acid is a similar polarity to the one that's being substituted? So we used to predict the pathogenicity of variants based on how well conserved it was through evolution, or how important the change was in, say, the amino acid sequencing, or the location of how it affected the protein. Is it in a highly conserved location or a really important functional location, such as binding of a cofactor, such as vitamin B6? But this is a higher level of sophistication using in silico computer modeling to really predict how these variants affect the protein structurally and functionally, how the protein folds, how it can function in vivo, at least theoretically in vivo. And by doing that, the investigators were able to come up with reclassifying many of the variants to understand which are pathogenic versus which are not. Most of these patients are misdiagnosed with cerebral palsy. And there's a big effort to take children with cerebral palsy of otherwise unknown etiology or unknown cause and see if they can be reclassified with a genetic disease. This is a huge area of research right now here in our department of neurology at Boston Children's, as well as all over the world. So if a child presents with a diagnosis of a significant motor disorder, especially a progressive one, not a static one, which would be non-progressive, but a progressive one getting worse over time, in the absence of a clear-cut cause that can be identified as, say, something that was pre-existing, prenatal, perinatal, or a known event postnatally. But in the absence of that, and even sometimes in the presence of that, a diagnosis of cerebral palsy alone should raise suspicion. In addition, these particular patients with AADC deficiency have a strong predilection to problems that are particularly associated with neurotransmitter disorders. For example, they have repeated oculogyric crises where the eyes will deviate up or off and to the side, and this can last for seconds to minutes to hours, during which they often appear very uncomfortable. And they're often misdiagnosed as having epilepsy because of the ocular deviation. But in the epileptic seizure, the ocular deviation is not as sustained and prolonged and tends to be associated with other more obvious stigmata of an epileptic event. So we're talking about oculogyric crises and other autonomic features in these patients: temperature instability, hypothermia, hyperthermia. Interestingly, a lot of nasal stuffiness. I know it might sound sort of common. Well, don't lots of kids have stuffy noses? But really severe problems with nasal stuffiness and drooling, excessive salivation, changes in heart rate that are unpredicted, and especially the oculogyric crises, as I mentioned, in the presence of other motor system problems such as dystonia. Dystonia, of course, is a sustained abnormal posturing of contractor and co-contractor muscles at the same time. Torticollis, a twisted neck, is a classic example of dystonia. So is writer's cramp, just to get an idea of what dystonia is. So they have the combination of dystonia with hypotonia. They tend to be very floppy. And these other autonomic signs I mentioned, oculogyric crises, and sometimes they get into problems with hypoglycemia. Remember, they can't produce catecholamine, so they may have a reduced catecholaminergic response, and that may affect their gluconeogenesis. Because AADC deficiency just represents the start of an avalanche of gene therapies that are about to come out for pediatric, neurogenetic, neurometabolic, and other diseases. And it's just absolutely amazing that gene technology was attempted 25 years ago in the case of ornithine transcarbamylase deficiency, a disease of the urea cycle disorder. And there were a lot of complications. It's a very famous case back in circa 1989, something like that. And the problem there was that the vector mediating the gene therapy, the AAV, the adeno-associated virus, caused liver failure. Since then, the technology has come a long, long way. And now there are a whole host of different vectors. The viral vectors, such as the one being used now for AADC deficiency, is AAV-associated virus, where it's an attenuated virus that doesn't cause infection of the liver. So the bottom line is the gene transfer technology has come a long way in the last 3 decades. And gene therapy may be administered intravenously, but then that raises the specter of immunologic rejection, sort of like you've heard of with bone marrow transplants. So that's a problem with IV. There could be intrathecal therapy, which means doing a spinal tap and injecting the gene into the subarachnoid space, which is being done for a very severe form of a metabolic epilepsy known as neuronal serial lipofuscinosis. And in this case of AADC deficiency, actually involves direct injection of the gene transfer into the brain, deep into the brain stem or into the basal ganglia, which are, as you can imagine from the terminology, basal ganglia, the deep nuclei or ganglia at the base of the brain, the basal. This is just the beginning of gene therapy that is going to become increasingly available for just a host of metabolic deficiencies where there's an enzyme deficiency and we're trying to replace the enzyme by either using enzyme replacement or better by injecting the gene to produce the enzyme. Enzyme replacement sounds very logical, but it's going to be a requirement for repeated enzyme infusions. This should be a one and done. This should be, we transfer the gene where it needs to go into the brain. The gene works, hopefully long-term, and there shouldn't have to be repeated administrations. Now, resources for this are going to be a problem. These are costly to develop. The price tags on these therapies are going to be so high, we have to figure out how to deal with them from the societal viewpoint, government, legislative, regulatory viewpoint. We have all kinds of challenges coming up, but this is really just the precipice of an avalanche of gene therapy for severe, progressive, life-threatening neurological disease. Is traditionally, it's been difficult to diagnose because it required a lumbar puncture or spinal tap, as known to most folks. And not only does it involve doing the spinal tap, but it's not routine studies that you could do just anywhere. It involves collecting the fluid in special tubes, immediately placing them on dry ice, having a perfect tap so there's not, say, blood that gets in the way. And then the dry ice has to be transported rapidly to one of only a couple specialized laboratories. There are only 2 in the United States that do these measurements. There are only a few throughout the world that do these measurements. So it has been a hassle. And this is a real impediment to diagnosis, requiring a spinal tap, because the blood studies, what we call peripheral studies, don't show these abnormalities in the neurotransmitters. This is a big challenge in neurotransmitter disease. On the other hand, with the mapping of the human genome, that really happened back in the 1990s and has since then gotten better and better and better and more accessible to all and more affordable to all. I mean, it used to be, even after the human genome was mapped, it would still cost $1 million to map someone's genome. Now we're talking about $1,000 just over the course of the last 25 years. So the widespread use of genomic sequencing has enabled us to discover diagnoses that we had no idea. I have seen a transformation in my career of seeing so many children with terrible neurological diseases and now having a diagnosis that's not just a descriptive diagnosis such as cerebral palsy or epilepsy or a certain type of epilepsy or autism spectrum disorder or a certain type of autism spectrum disorder or a syndrome named after the doctor who described it or the first child who was described to have it. But now we are really making specific, pinpoint, personalized diagnoses based on genomic sequencing. This has opened an entirely new vista in our field and opens us to the possibility of specific genetically targeted personalized therapies, whether it involves enzyme replacement therapy, gene therapy, antisense oligonucleotide therapy, or other maneuvers affecting the RNA and not necessarily the DNA, but editing of any of those that has just burst upon the scene. We're going to have to figure out how to deal with all this, both as clinicians and based on society and resources. This paper, this approach that we use to study AADC deficiency is about to go wildfire and be spread to all these diseases that we're now identifying that we never knew about before. To have seen that in my career has been astounding.